The meselson-stahl experiment aimed to investigate the mode of DNA replication, especially whether it is followed a conservative, semi-conservative, or dispersive pattern. Here, I will be giving the detailed explanation. Okay, so the first step is isotopic leveling. Actually, isotopic leveling. The researchers grew E. coli bacteria in a medium containing the heavy isotope of nitrogen N15, okay, for several generations. So this resulted in all the DNA in the bacterial cells incorporating this heavy isotope. So here you can see the N15 containing medium is there, and E. coli cultures are grown in this heavy isotope. So then they continue growing the first generation in N14 medium. So the second step, what is harvesting the DNA samples? Harvesting DNA samples. After several generations, the bacteria transferred to a medium containing a lighter isotope N14. This shift to a lighter isotope would allow for detection of any new synthesis. Okay, so here you can see the DNA composition is heavy DNA, that is N15 N15. Okay, but when the bacterial samples were transferred to the N14 containing culture, here you can see the intermediate density DNA N14 N15. The extracted DNA was actually subjected to a density gradient centrifugation. It is a technique that separates molecules based on the density. A cesium chloride gradient was used, which forms a density gradient when spun in a centrifuge. So, from this cycle, you can observe that the DNA samples were analyzed based on the buoyant density. Conservative replication would result in two distinct bands, one heavy, one light. That is semi-conservative replication would show an intermediate band. So here you got the intermediate band. This shows what? This shows the semi-conservative nature. Semi-conservative nature of DNA, because it is a intermediate, because it contains one strand with N15 level and one other strand with N14 level. So you can see that after one round of replication in the N14 medium, the DNA exhibit a single band of intermediate density. This finding supported the semi-conservative mode of DNA replication. Additionally, the subsequent rounds of the DNA replication in N14 medium confirm the persistence of the intermediate band. You see here also you got the N14 N15. If you grow in another N14 containing medium, here also you got this intermediate band N15 N14. So you can see it is consistent with the, as we expected, that is semi conservative application. So, in summary, the meselson-stahl experiment used the isotopic leveling, density gradient centrifugation, and careful analysis to demonstrate that DNA replication follows a semi-conservative model, with each new DNA molecule containing one parental strand and one newly synthesized strand. Here you can see there is one parental strand and another is the newly synthesized strand. So this confirms the semi-conservative mode of replication. Thank you.